So we are entering a new topic, the functions of the DCT, the reabsorption of the solutes and the water. The DCT is essentially the extension of the thick ascending limb of the loop of Henle. Hence, physiologically, it is close to the thick ascending limb of the loop of Henle. Therefore, the DCT is relatively impermeable to the water, whereas the solutes are transported. As the solute is removed in excess of the solvent, further dilution of the tubular fluid occurs in this part of the nephron. In the initial segment of the in the initial segment of distal tubule, sodium and the chloride are reabsorbed from the tubular fluid into the tubular cells by Na plus Cl minus transporter symporter, which can be seen over here. Next, the sodium is removed from the tubular cells into the interstitial space by the Na plus K plus pump located on the basolateral membrane, and Cl minus is removed by the Cl minus channels, which can be seen over here. However, in spite of the osmotic gradient across the tubular epithelium, water reabsorption is poor. Only about 5% of the filtered water is removed in the DCT. Thus, dilution of the tubular fluid that begins in the thick ascending limb of the loop of Henle continues into the DCT. Thiazide diuretics acts on the DCTs, Na plus Cl minus co-transporters. Thiazide diuretics such as chlorothiazide and metholazone diuretics act by inhibiting the Na plus Cl minus co-transporter in the early part of the DCT. This causes the natriuresis and the diuresis. Next, coming to the functions of the collecting duct. It is not a part of the individual nephron. As the collecting duct is a terminal part of the nephron, it is its ability to alter the transport of the solutes in the water contributes considerably to the final osmolality and composition and volume of the urine. Most of the excreted potassium ions in the urine come from the collecting duct. The transport process in the collecting duct are also influenced and fine-tuned by the hormones such as the aldosterone and the ADH. Now, the reabsorption of the solutes in the collecting duct. Epithelium of the collecting duct has two types of cells, which include the principal cells and the intercalated cells. Principal cells reabsorb the sodium ions and secrete the K+. How to remember this? Sodium includes A, it's something related to absorb, so reabsorbs. Intercalated cells secrete either H plus ions or the HCO3 minus ion. Principal cells also contribute to the K plus metabolism, and intercalated cells play a role in the acidification of the urine. Intercalated cells also reabsorb the bicarbonate and the potassium ions. Coming on to the principal cells help in the reabsorption of the sodium ion and secretion of the potassium ions. This depends on the sodium potassium pump. K plus excretion in the collecting duct mainly occurs in the cortical part. Now, the mechanism of the sodium reabsorption and the K plus secretion in the principal cell of the collecting duct can be seen over here. The aldosterone increases the activity of the epithelial sodium channel, that is ENaC, that increases the sodium reabsorption and stimulates the activity of Na plus K plus ATPase that helps in generating gradient for the Na plus absorption and K plus secretion, which can be seen over here. Aldosterone acts on the nucleus, which leads to the production of proteins, that is SGK and other proteins. These proteins are involved in the two function. One, they help in ENaC production, which we are discussing here. That is the role of the aldosterone. The sodium is reabsorbed from the tubular fluid by the principal cells by diffusion of the sodium channels in the apical membrane of the epithelial cells. These sodium channels are known as the epithelial sodium channels. Aldosterone facilitates the genomic activity to produce the SGK and the other proteins that form the ENaC in the cells and their insertion into the luminal membrane. The sodium ion that enters cell in the pumped out the sodium ion that enters the cell is pumped out into the interstitial space by the Na plus K plus pump, the activity of which is promoted by the aldosterone, which can be seen over here. This SGK and other proteins, they have two roles. One is ENaC and other acts on the Na plus K plus ATPase. ENaC increases the sodium influx from the tubular lumen into the cells, and Na plus K plus ATPase helps in sodium movement from the tubular cell into the interstitial fluid and absorption of the K plus from the interstitial fluid into the principal cells of the collecting duct. Therefore, aldosterone acting on the collecting duct increases the sodium absorption and facilitates the K plus excretion. The hypernatremia and the hypokalemia are the features of the hyperaldosteronism. The K plus sparing diuretics. K plus sparing diuretics include the amyloid and spironolactone. Inhibits the sodium reabsorption from the collecting duct by antagonizing the effects of the aldosterone. Amyloid inhibits ENaC and spironolactone inhibits the Na plus K plus pump. Inhibition of the Na plus reabsorption decreases the magnitude of the negativity in the lumen. This facilitates the chlorine reabsorption by the paracellular pathway. Because it decreases the negative charge in the lumen, it also decreases the K plus secretion into the tubular fluid. Thus it decreases the K plus excretion in the urine. Therefore, they are called as K plus sparing diuretics. This is usually used in the patient with increased K plus excretion in the urine. Next, coming on to the intercalated cells. These cells are scattered among the major cells in the collecting duct. On the luminal side, the membrane contains the H plus K plus ATPase that pumps H plus into the cell and K plus into the tubular fluid, which can be seen over here. K plus into this diagram is basically wrong. H plus into the tubular fluid and K plus into the cell. Next, thus intercalated cell contribute to the acidification of the urine and conservation of the K plus. Reabsorption of the water. The reabsorption of the solute and the water from the collecting duct mainly depends upon the efficacy of the ADH acting on it. In the cortical part, ADH increases the permeability of the collecting duct to water by inserting the aquaporin 2 channel spatially on the luminal membrane of the principal cells. This mechanism can be seen over here. ADH increases the CAMP. ADH bind to the V2 receptors of the epithelial cells of the intercalated epithelial cells of the collecting duct. ADH bind to the V2 receptors over here. The CAMP stimulates on binding, it converts the ATP into the CAMP, which further CAMP activates the protein kinase A, which promotes the insertion of the vesicle containing aquaporin 2. This can be seen here. Activate the protein kinase A and increases the vesicle containing aquaporin 2. That increases the water permeability. This causes an increase in the water permeability. Next, the CAMP also increases the gene transcription that increases the formation of the aquaporin 2. CAMP acting on the nucleus stimulates the gene transcription, which increases the aquaporin 2 formation. In the presence of the ADH, water moves out of the hypotonic tubular fluid into the cells of the cortical collecting duct and then from there into the interstitium of the cortex. This helps in the reabsorption of the 10% of the filtered water in the tubular, and the tubular fluid becomes isotonic. This isotonic fluid then moves into the medullary portion of the collecting duct. Here, osmolality of the interstitium is very high. Water reabsorbed from the tubular fluid into the hypertonic medullary interstitium by the osmotic effect, which accounts for 5% of the water reabsorption. This play important role in the countercurrent mechanism and makes the urine concentrated. In the presence of the adequate ADH, osmolality of the urine may reach to the 1400 milliosmol per kg of the water. In absence of the ADH, cells of the collecting duct are relatively impermeable to the water. However, collecting duct is not totally impermeable to the water, and therefore even in complete absence of the ADH, about 2% of the filtered water is reabsorbed. Coming on common case of diabetes insipidus, this is a condition of the excess urine formation that occurs due to the ADH deficiency. It is called as the neurogenic diabetes insipidus or failure of the collecting duct to respond to the hormone. The nephrogenic form, which is X-linked recessive type, occurs due to the mutation of the gene for V2 receptors.